Hello, so this is the third part of lecture 1. Okay, so let's start. So you already know that most of the enzymes are proteins except a small group of catalytic RNA molecules which are called ribozymes. Okay, and the catalytic activity of the enzymes mainly depends on the integrity of their native protein conformation. Okay, so if this structure is denatured or dissociated into its subunits, enzymes lose their catalytic activity. Okay, so that's the primary, secondary, tertiary, and quaternary structure of protein enzymes are essential for their catalytic activity. Okay, and like the others proteins, enzymes have molecular weights ranging from about 12,000 to more than 1 million Dalton. Okay, so some enzymes they can work by their own. I mean, একাই একশোটাই বাকি. Okay, so some enzymes they don't require any other chemical groups for their catalytic activity other than their amino acid residues. I mean, their amino acid residues are good enough to do the catalysis. catalysis. However, other enzymes they require help. They require an additional chemical component for their activity. Okay, so and these additional chemical components are basically two types: cofactor and coenzyme. So cofactor, they are either one or more inorganic ions such as this iron 2 ion, magnesium 2, manganese 2, zinc 2. Okay, these are cofactor. And coenzymes, they are is a complex organic or metal organic molecules. Okay, so many enzymes actually they need help from the cofactor and coenzyme. Basically, this cofactor and coenzyme support them by providing additional chemical groups. Okay, so anyway, so a coenzyme or metal ion that is very tightly or even covalently bound to the enzyme protein is called a prosthetic group. Okay, and a complete catalytically active enzyme together with its bound coenzyme and or metal ions is called a holoenzyme. And the protein part of such enzyme is called the apoenzyme or apoprotein. I mean, the protein part of the enzyme itself is called apoenzyme, and when this apoenzyme is bound with its coenzyme or cofactor, it is called holoenzyme. Okay. This slide it shows some examples of coenzyme and metal ions, that is cofactors, and their related enzyme. For example, pyruvate dehydrogenase. These enzymes require thiamine pyrophosphate. Okay, and carbonic anhydrase. This enzyme need zinc 2 ion for the as a cofactor. Okay, so okay, you don't have to memorize all these things. So you can just have one or two will be good enough. Okay, you don't need to memorize. This is tough. I can understand. Anyway, so and the next table, this one actually also shows some example of the coenzyme, and also it shows that what kind of chemical groups they transfer during the catalysis. For example, coenzyme A. Okay, it can transfer acyl groups. Okay, so again, you don't have to actually memorize these things. Anyway, so now let's talk about nomenclature of enzyme. Okay, everything need a name, right? We all have at least some name. Actually, everything has a name. Without name, it becomes very confusing, right? So it's also true for the enzyme. We should assign some name to the enzyme. Okay, so usually many enzymes they have been named by adding the suffix "ase" to the name of their substrate or to a word or phrase describing their activity. Okay, for example, urease. Okay, urease is an enzyme. It's named such because it do hydrolysis of urea. This is the catalysis. Okay, so this is the catalysis. So urease they catalyzes hydrolysis of urea. So that's their function, their activity. They hydrolyze urea. So that's why the name of this enzyme was urease. We just added the suffix ase. Okay, and another example is DNA polymerase. This enzyme ase. Okay, DNA polymerase. So this is an enzyme. So DNA polymerase catalyzes the polymerization of nucleotides to form DNA. Okay. However, before knowing the specific reaction that are catalyzed by enzymes were named mainly by their discoveries. Okay. For example, uh, the enzyme pepsin. Okay, it was named so because it was discovered from the digestion. I mean, what I wanted to mean that we know that this enzyme they can digest food, and this pepsin enzyme they can digest food. Okay, and in Greek there is a word pepsis. Pepsis means digestion. So from this the name pepsin came. Okay, as this enzyme can digest food, its name is pepsin. Okay, and another example is lysozyme, which was named for its ability to lyse the bacterial cell walls. Okay, lyse means break down. Okay, so still some other enzymes are named for their source. I mean, some enzymes they are named because of their activity, some from their discovery, some from their source. For example, trypsin. Okay, it's named in part from the Greek word trine, which means to wear down. Okay, was obtained by rubbing pancreatic tissue with glycerin. I mean, they rub the pancreatic tissue with glycerin and get the enzyme. So that's why they it was named trypsin, because in Greek, trine name to wear down. Okay. So, as there was no uniformity in the naming, sometimes sometimes it was like that. Sometimes the same enzyme has two or more names. Okay, or two different enzymes can have the same name. This is a problem. Like there is no uniformity. One particular enzyme it may have several names, and two different enzymes may have the same names. This create confusion. So to overcome this ambiguities, and also as the number of newly discovered enzymes are increasing, an international agreement was done. Okay, and a system was adopted for naming and classifying enzymes, a uniform systems. Okay, basically this international agreement was done by an international organization. Its name is the Enzyme Commission. Okay, so the Enzyme Commission system, they divides enzyme into 6 classes, and each of these classes are subdivided into subclasses based on the type of reaction catalyzed. This table shows the 6 classes of the enzyme and their associated functions. So these are the 6 classes: oxidoreductase, transferase, hydrolase, lyase, isomerases, ligases. So oxidoreductases, they do the oxidation reduction reaction, and they do this by transfer of electrons. Okay, and transferase, okay, they transfer groups. Hydrolases, hydro water, they do hydrolysis reactions. Okay, transfer of functional groups to water. Lysis means breakdown. So they cleave different bonds, like they cleaves of carbon carbon, carbon oxygen, carbon nitrogen, or other bonds by elimination, leaving double bonds or rings, or addition of groups to double bonds. In short, lysis, they lyse bond. Okay, they break down bond. Isomerases, they transfer of groups within molecules to yield isomeric forms. Isomerases, they change the isomeric form. Okay, and ligases. So what ligases do? They form bond. I mean, lysis, they break down bond. Ligases, they form new bond. So formation of like carbon carbon, carbon sulfur, carbon oxygen, and carbon nitrogen bonds by condensation reactions coupled to cleavage of ATP or similar cofactor. Okay. Uh, uh, so uh, and these were the 6 classes of the enzyme, and according to the international agreement, each enzyme is assigned a 4-part classification number. I mean, to name the enzyme, each enzyme is assigned a 4-part classification number. Okay, and a systemic name which identifies the reaction it catalyzes. So okay, let's talk about the example. Consider this reaction: ATP plus D-glucose uh, turn into ADP plus D-glucose 6-phosphate. Okay, so this reaction is catalyzed by enzyme which name is ATP glucose phosphotransferases. Okay, and from the reaction actually we can see that ATP triphosphate. From here, one phosphate group was transferred into D-glucose. That was it became D-glucose 6-phosphate, and the ATP it's lose one phosphate, so it becomes ADP, diphosphate. Okay, so that means a phosphate group was transferred in this reaction, and this transfer of phosphate group was done by this enzyme ATP glucose phosphotransperases. Okay, so according to the international system, the enzyme commission number of this enzyme ATP glucose phosphotransperases is 2711, 4 part. Okay, and each of this part has a specific meaning. So what are those? The first number 2, it denotes the class name. 2 means 2 belongs to the transferase, right? 2 number 2 is transferase, so it's a transferase enzyme. The second number denotes the subclass 7, and 7 denotes refers to the subclass phosphotransferase. And the third number, which is here 1, number 1. Number 1 denotes that a phosphotransferase with a hydroxyl group as acceptor. Okay, and the fourth number, it denotes that the D-glucose as the phosphoryl group acceptor. Okay, so every enzyme, based on the enzyme commission, every enzyme has a 4-part name, basically 4 number, and each of number has a specific meaning, and this 4 number can identify that particular enzyme. Okay, so that's all for this lecture. So in the next lecture, we'll talk about the types of specificity of enzyme. Okay, thank you.